Hi, everyone. So let's give some more minutes. People tend to log in right after 10, so we'll wait a couple more minutes. Thank you. Okay, I think we can get started. So thank you, everyone, for joining us today. We have another fantastic speaker, Rosie Paterno. She got a medical degree from University of Palermo, Italy, and then obtained residency in anesthesia and critical care from University of Milano. And she did first postdoc training in Akiva Cohen's lab at Children's Hospital of Philadelphia, and currently in Scott Barban's lab at UCSF. Today, she will talk about hippocampal oscillations, CNT and AP2 mice, and middle ganglion eminence. Rosie, please take it away.
Thank you very much for the introduction and also for the opportunity today to talk about a part of my work that I have done in Scott Barban Lab. As you say, I'm going to talk about an animal model today, the KNAP2 mice, which is an animal model for autism, hippocampal oscillation, and the MG cells, or the medial ganglionic eminence cell. In general, my overall goal is try to understand how a brain disease will affect the underlying circuit, focusing in particular to the hippocampus. Using animal models, I study how the brain disease will affect the hippocampal circuit, in particular while the animal are performing different type of memory tasks. The goal is to identify specific electrophysiological signal and study the hippocampus and the circuit in detail. Then to apply specific type of tools that it can be cell transplantation, for example, to rewire the circuit, or closed loop deep brain stimulation to retune the circuit, and therefore to precisely modify the dysfunction. All those tools then can be applied to the human, so it has a translational purpose. Today, I'm going to talk for most of my talk about autism and the hippocampal network dysfunction. And in the last 10 minutes, I will focus to the MG cell transplantation as a tool that we can use to rewire the circuit, or as a therapy in some other cases. Focusing on the autism. Autism is the autism spectrum disorder is a neurodevelopmental disorder. And in the last 20 years, since 2000, has been shown that there has been increased incidence of this disease. In particular, 1 every 100 children is affected. Despite the fact this is a disease that presented with a different wide range of defect, all the features can be classified in 2 main cores. Those children, they have alteration in the social interaction, but also they are characterized by a stereotyped movement in the sensory and involving the sensory and the motor circuit. But also they have a wide range of cognitive dysfunction. Today, there is not a therapeutic approach that target autism spectrum disorder. The only FDA-approved drugs, they target co-occurrent symptoms like agitation, hallucination, and therefore they are used atypical antipsychotic, which have in a long-term side effect. So many studies have tried to identify the cause of autism, and there are so many studies focusing on the genomics and trying to understand if there is an association with some genes, and so many different genes have been identified. But the key is to try to understand if there is a common pathophysiological hallmark, given the fact that so many different genes are involved. This is a paper from 2009, so it's already more than 10 years ago, in which they advanced the hypothesis that the complexity of autism spectrum disorder can be due to the fact that there is an alteration in the excitatory-inhibitory balance during a critical point during development. And in particular, the interneuronal population has been identified as participating in a key role in determining this imbalance. A specific local type of circuit study have identified between all the interneurons, parvabum interneuron cells has a key cell in human and also in different animal models have been shown that these parvabum interneurons are altered or reduced, as we can see here, so many different animal models. Given the fact that autism is a neurodevelopmental disorder, this is a very simple and schematic way to see how important interneural development is and how it will change the circuit. In particular, interneurons are born already during the embryonic day 10 or 11, and they need to migrate, they need to reach their final brain structure when they participate to the maturation of the circuit. So they have a fundamental role. And in particular for parvabum interneurons, which are a key player in this type of disease, we can see that their maturation is very slow. We have a full mature parvabum interneurons only at P30. A full completed maturation. Given the fact that these interneurons are found across and throughout the brain, it's not surprising that different brain regions are involved in the autism spectrum disorder. As we can see here, different regions have been studied going from the striatum to the cortex and the cerebellum, including the thalamus, but also the hippocampus. Even though so many different regions have been involved, the studies about how the hippocampus is involved are just few. The involvement of the hippocampus in autism spectrum disorder also is supported from human studies. In recent research have highlighted that the subject with autism also present a memory deficit, and in particular, they have a decreased success in memory retrieval. This is a study from 2017 showing using fMRI in subjects with autism while they were performing memory tasks. And they were studying the connectivity in the memory system. And in particular, they found that there is a decrease in hippocampal connectivity during memory retrieval. not only the memory system is involved, but also in this subject, there is 530% that has as a comorbidity epilepsy. So all those evidence together suggest the hippocampal involvement in autism spectrum disorder. The question is, what is the mechanism, how the circuit is involved, and how it changed from normal condition? To answer that question, we use a KNAP2 animal model, and I will later explain why we chose this animal model. We run a battery of tasks to confirm the autism phenotype in this animal model, but because we were interested in the hippocampus alteration, we used the hippocampal dependent memory task. We recorded the local field potential across the different layers of the hippocampus using silicon probes. And then, in addition, we run some in vitro experiment and immunohistochemistry to understand the involvement of the parvamine interneurons in the circuit. The KNAP2 gene is a gene that has been known for a long time, but has been associated to autism because it has been identified in a group of children which present with autism phenotype, but also epilepsy. This is a gene that produces a protein, which is the Caspr2, and it's known for clustering voltage gate potassium channel. However, this protein is also highly expressed during development, and in particular in area they are involved with the formation of interneurons. Therefore, there is also an involvement in the inhibitory component during development. It is also highly expressed in hippocampus. It has been shown to show a decrease of parvamine interneurons in different brain regions, including the hippocampus, but also a defect in the performance with the radial maze of worry motor maze. So we first ran a battery of tests to confirm that this animal model had the phenotype. So as in literature, we could replicate most of the finding already present in literature, where the animal will spend more time in the open arm using an elevated plus maze. We did not find any difference in social interaction using this modified social three-chamber social test. They had an increased stereotype behavior using self-grooming, and also they were hyperactive using open field test. But the question was how the hippocampus involved. Can we identify any difference in the behavior performance using hippocampal dependent memory task? And we chose the special object recognition task. This task is running in 2 days. The first day, the animal is exposed to the open field, completely empty. And the second day, the animal is exposed to 2 identical objects. After a delay of 2 hours, one object is moved in the new location, and the normal animal will spend more time with the object that was moved, recognizing that there was a change in the space. Despite the fact that those animals were hyperactive, also in different tasks, it's important to notice that there was no difference in how much time they spend with the object. Control and KNAP2 were absolutely normal. However, during tests, the KNAP2 animals failed to discriminate the object that was moved in a different location, as we can see here. To identify if that was a specific type of alteration space memory in the hippocampus of these KNAP2 animals, we also run the object congruence task. This object congruence task, which basically tests if the animal is able to differentiate between a different type of context without the involvement of space, we did not see any difference, meaning that the KNAP2 animals were able to differentiate different contexts. So in summary, and that from the behavior standpoint, what we found is that the KNAP2 animals in our hand has most of the feature of the autism spectrum disorder, but also have a reduced spatial discrimination ability, which is a type of hippocampal dependent behavior, but has a normal context discrimination ability. To study the neural network activity, we implanted electrodes, and this is an example of the electrodes that we used. Those are the silicon probes in which there are many different electrodes in each shank in order to record the local field potential across the different layers of the hippocampus. After 1 week of recovery, we record the animal while it was in the home cage. And in particular, for 5 days in a row, and we record it for 1-2 hours a day. The analysis that I'm going to show here is focusing on the last 3 days to remove any contamination, any anxiety coming from the new recording system and the new room where the animal is recorded. After the recording in the home cage, the animal, we recorded open field task. In particular, we recorded the animal for 3 days in a row, and each day we use different open field tasks, which are characterized by different type of bottom and walls, and so each day is a completely different task. After the experiments were done, we perfused the animal to identify the electrode's location. However, to precisely identify the electrode location with these silicon probes, not only immunohistochemistry has been used, but also different techniques, features of the local field potential to precisely identify the electrodes. Focusing on the open field, we first want to understand how the network activity is performing during navigation. And so we use the open field exposure. What we can see here, this is an example of the animal moving around in the open field, and we can see that both condition control, like NAC2, they will move around with no problems. However, the NAC2 animals, they will be more active, and therefore their hyperactivity is still preserved despite the fact that they had surgery or there is a recording system. So the behavior is preserved. This is an example of while they were performing the behavior, we recorded local field potential. And this is an example of the silicon probes that I used. And you can see here how we can record across the different layers of the hippocampus. And this is an example of the local field potential across the different layer, in particular the CA1 area, stratum pyramidale, radiatum, and lacunoso molecolare, but also the dentile gyrus. And from the local field potential, it's important to notice by eye already how different the local field potential is, because each layer has a different, carrying different information. To precisely understand, to precisely interpret the local field potential which is recorded across the different layers, I am referring to this paper, which was published in 2017. But it's a paper in which they tested and investigated the upstream region input to the hippocampus and how the local circuit will modulate that. And at the end, they also developed a model, which is called entorhinal cortex CA3 dual input model, in which most of the findings in the field could be explained by this model. In a schematic way, in particular, the gamma oscillatory activity across the different layers of the hippocampus, and studying those, they give information about the upstream region. In particular, entorhinal cortex, the entorhinal cortex input arrived to the stratum lacunoso molecolare, and this is a recording as a mid-gamma activity in the frequency between 60 and 90 hertz. The CA3 input instead arrives to the stratum radiatum and is recording as a slow gamma in the frequency range between 30 and 55 hertz. And the activity in the response from the stratum pyramidale, therefore the firing and when, is recording as a fast gamma with a frequency more than 100 hertz. This is the structure of the hippocampus, but this is to interpret the result about the different the power spectrum across the different layers of the hippocampus. And we in particular focus on the slow gamma and the mid gamma to understand the input from CA3 and entorhinal cortex. And what we found is that across the different layers, we can see that we found a decreased mid-gamma activity in the 60-90 hertz in stratum pyramidale and stratum lacunoso molecolare. But also we found an increase in slow gamma activity in stratum, statistical significant in stratum lacunoso molecolare, and a slightly increase in stratum radiatum. So as a summary of these first findings, we have a decreased input from entorhinal cortex and increased input from CA3. The entorhinal cortex hippocampal system not only relate to the rate code, but also relate to a phase code, which is related to the phase preference of theta and gamma oscillations. In particular, this is a theta rhythm. This is a theta rhythm oscillations are recorded in the hippocampus, and they are the main oscillatory activity in the hippocampus. The theta cycle is thought to be a temporal window in which the input from the upstream region, so CA3 and entorhinal cortex, through gamma activity will be higher at preferred theta phases. In particular, slow gamma will be higher at the descending phase of the theta cycle. Mid gamma will be higher at the peak of the theta cycle, and fast gamma will be higher at the trough of the theta cycle. So when we look at control animals in this case, this is a distribution of the power of gamma oscillation across the theta cycle. So this is an example of the theta cycle going from minus pi to plus pi, so trough to trough. And here we can see that there are three frequency bands: the slow gamma, the mid gamma, and the fast gamma. And as expected, we can see that we have an increase of slow gamma activity at the descending phase of the theta cycle. We have an increased mid gamma activity at the peak of the theta cycle, and an increase of the fast gamma activity at the trough of the theta cycle. So this is as expected in control animals. When we compare our findings here with CNAP2, we can see that the three frequency band, so the three different gamma, slow, mid, and fast gamma, they have the same distribution and the same theta phase preference: slow gamma descending phase, mid gamma at the peak, and fast gamma at the trough. However, we can see how the mid gamma modulation is reduced than control animals by eye. To confirm our finding, here we can, this is a cycle by cycle analysis and the statistics to confirm that in the mid gamma range between 60 and 90 hertz, in the theta cycle, which goes from minus pi to plus pi, we have a decreased mid gamma power at the peak of the theta cycle. Therefore, the modulation is reduced. All those findings were located at the pyramidal cell layer. To confirm that the same findings are coming from the stratum lacunosum molecolare, this is where the mid gamma activity is, the input of the mid gamma activity is arriving, we did a current source density analysis, which were able to isolate the mid gamma activity. As we can see here, we predominantly have mid gamma activity, and this mid gamma activity is higher at the peak of the theta cycle. But when we compare to CNAP2 animals, we can see how the modulation is way reduced. There is a decreased mid gamma power at the peak of the theta cycle, also in stratum molecolare, in stratum lacunosum molecolare. So if we want to summarize the results up to now, we can see that in stratum lacunosum molecolare, we have a reduced mid gamma power. We also have a reduced theta mid gamma modulation, and a slightly increased slow gamma in the system. We recorded also the animals while they were in the home cage, and in particular during prolonged period of immobility in the hippocampus. One of the most important electrophysiological signature is a shockwave ripple. Shockwave ripples are characterized by a depolarization wave, or called sharp, in the stratum radiatum. That responds an input coming from CA3. And the stratum pyramidale will respond with a really fast oscillatory activity, which is called a ripple, and it goes from 150 to 150 hertz. The important part about the ripple is that this is a cognitive features because it's involved in episodic memory, but also in planning. The reason why it has such an important function is because of the content of the ripple activity. In particular, if we look at the ripple content, which is represented here, we can see that the ripple represents a neuronal sequence that is temporally and spatially precise and has a content and information, containing an information. The sequence is a predetermined type of sequence, which represent the same sequence that wolves learn during a wake state, during performing tasks. What we found is that there is a reduced sharp-wave ripples power in KNAP2 animals. Here in the left, we calculated the peak of power during the event, and we are averaging here around 10,000 events in each condition. And we can see that there is a difference in the peak of power. But also, we can see that when we average all the events in each session, the power spectrums has the same feature. There is a decrease of sharp-wave ripples power. All the other features of sharp-wave ripples power were normal, but I want to point out in particular the fact that there was no difference in the depolarization wave coming from CA3. Therefore, when we interpreting our results, we think that in addition to a reduced mid-gamma power and theta-gamma modulation, in the stratum pyramidale, we have a specific sharp-wave ripples power decrease, which mostly likely depend on the alteration of the interaction between pyramidal cells and interneurons. Parvalbumin interneurons, so we remember that has an important function in the maintenance of the sharp-wave ripples, highlighting the fact that most likely there is a decreased pyramidal cell synchronization. The animal model that we are studying also has a pathophysiological underlying mechanism in which inhibition has been identified as the driving force. And in particular, we want to understand the inhibitory neurotransmission system in area CA1 of the hippocampus. So what we using in vitro technique, we are recording pyramidal cells, and we in particular recording spontaneous inhibitory postsynaptic input, spontaneous iPSC. And what we found is, as expected, we found a decrease of frequency and amplitude in spontaneous iPSC, as expected. But also therefore, there is a decreased inhibitory neurotransmission. There is an alteration in the inhibitory neurotransmission. To confirm if that was dependent upon parvalbumin interneurons, we used the EpiVT tomato line. And we want to study the density of parvalbumin interneurons in the hippocampus. So using this line, we identified that there was a decreased density of parvalbumin interneurons in the hippocampus, has been shown in other studying literature. But this decrease in parvalbumin interneurons was mostly driven by area C1 of the hippocampus. So, area C1 of the hippocampus. So, as a summary of this first part of the talk, KNAP2, what we found is that the KNAP2 animal model has an alteration in the ability to discriminate spatial memory, at the which is hippocampal dependent. At the hippocampus, we had a decrease of sharp-wave ripples specific in the pyramidal cell layer, but also a reduced theta-gamma modulation. All this was correlated with a reduced density of parvalbumin interneurons in area CA1, and also a reduced inhibition in pyramidal cell. If we want to interpret, we want to put together all those findings and understanding and give a possible interpretation of what is happening in the KNAP2 animal model, we go back to our model. So the CA1 area is, the circuit is a very dynamic type of circuit. And in particular, the CA3 and the entorhinal cortex input, they are competing type of input. What means is that the strength of the one input is prevalent than the other input in some time points, and vice versa. Using the model, the entorhinal cortex dual input model, and we can see that this input coming from upstream region, and in particular the strength of the input, which is recording with mid gamma and slow gamma, is depending upon two components: the upstream region, but also the local activity of the CA1 area. So the strength, and in particular, the strength of the entorhinal cortex, which is recording with mid gamma, is higher when there is an increase of perisympathetic inhibition and a decrease of dendritic inhibition. The CA3 input, instead, the strength is higher when the perisympathetic inhibition is reduced and when the dendritic inhibition is higher. Seems that there is a control, the local inhibition is able to control the strength of the input from the upstream region. What we think is happening in a possible explanation about this animal model is that the input coming from entorhinal cortex is lower, and therefore the stratum pyramidale, CA1, is more tuned to CA3 input in association with the fact that we have a reduced perisympathetic inhibition. We, of course, need to remember that this is the autism is a neurodevelopmental disorder, and so different brain regions can be involved. So as a future direction, we could also understand if what we see in the CA1 area is just a local alteration of the circuit, or in addition to the fact that we have an alteration of the local circuits, as we have seen, there is an alteration in the upstream region, and this is as a future direction. But also we can see if these findings here are common and can be extending to different animal models as a common pathway, despite the fact that different genes are involved. And third, but this is a broader future direction to understand if we can identify during development a specific time point in which the circuit deviate from the normal, and in that point interfere, precisely to understand where. With this one, I will conclude the first part of my talk. The second part will instead focus to MG cell transplantation as a possible tool of therapy. As I said, the overall goal that I have is that I would like to precisely modify the circuit, and because the circuit needs to be modified differently, I think it's worthy to understand if we can also use MG cell transplantation and establish a way that can be used as a precise type of medicine. MG cell transplantation. To talk about the technique, I will introduce first the MG cells. So we know that in the interneurons are one of the largest is a larger population. They represent 40% of the neural population in the cortex. And there are so many different subtypes of interneurons. Only in the hippocampus we can almost count 20 different ones. But they have a characteristic during development because they are originating from two precise progenitor zones, which are called the medial ganglionic eminence, the MG, and the caudal ganglionic eminence, the CGE, and in small amount from the preoptic area. The characteristic, they originate already embryonic day 8, 9, and at the 8, 13.5, they start migrating. So the way that they migrated, those are progenitor cells, they migrate tangentially in a long range to reach their final position. This migration is a very, very complex system where so many different components will play a role to make sure that they will migrate correctly. The migration is also a long process because it was started in a late embryonic developmental developmental time, and will continue during the first couple days of after the animal is born. Those cells is important. They are able to integrate, so they will form synapses with the circuit, and they will maturate in what we know they are GABA neurons. In particular, the interneurons can be classified depending upon if they origin from the medial ganglionic, the MG, the medial ganglionic eminence, or the caudal ganglionic eminence. From the medial ganglionic eminence, parvalbumin interneurons, somatostatin, and NOS origin from MG. For the CG, we have instead VIP called retinine and reelin, which have different properties. When we use MG cell transplantation as a technique, this was already established more than 20 years ago, where the technique consists in, in taking the developmental brain in the embryonic day 13.5, in dissecting the cells from the MG area. Those cells will be dissociated and it will be injected in the recipient mouse. And the injection, after the cells are injected, the cells will migrate from the injection site and reach in a different part of the brain region where they were injected. The migration, we remember, it's a pretty fast type of migration because after 7 days post-transplantation, they must have already reached the final structure where they will be. Important is that these transplanting MG cells, therefore, they have the migration property when they are transplanted, but also they have the ability to integrate in the circuit. This is a paper from 2017 from our lab showing that transplanted interneurons in the circuit, which are represented here in green, using optogenetic combination of technique, optogenetic equianimal and patch clamping, we were able to demonstrate that those cells actually form active connection with the native pyramidal neurons. So this type of connection is active. So those cells are actually functioning and integrating in the circuit. And important also that not only are integrated, but also they maturate in the interneurons that in particular, they will maturate in somatostatin interneurons and parvalbumin interneurons. And numerous studies, including these from 2014, have shown that these cells, the MG cells already maturating in the host brain, will have the features of the mature interneurons, as been seen here. They are fast spiking and regular spiking non-pyramidal interneurons. So this type of technique has been extensively used in literature and has been used in physiological condition, in particular in pups, because it has been used to study the property of the MG cells, how they migrate, how they integrate, and so on and so forth. But also have been used in different disease animal model as a tool or a therapy to modify the circuit. Pretty successful because they actually can rescue different types of outcome. But the important part is because I am interested in rewiring the brain and I want to know precisely how I can actually use this technique to precisely modify it, I noticed that in literature, there is a wide range of protocol that they've been used. The number of cells injected, it can go from 15,000 to 1.5 million. But more important is that there is a difference in how many cells are going to survive in the brain circuit that we are studying. And more important, how there is a difference in how much they are migrating. So we think that the MG cells transplanted, they have their feature to migrate, integrate, and maturate, has been shown in pups. But the recipient animals also play a role and may change some of these features. So to test that, in particular, we want to control two different factors on the recipient animals. One was the brain region, and the other one was the age. Do we have a difference depending upon the brain region where we are going to transplant, or do we have a difference depending upon the age of the animal? And for age, we transplanted in pup, juvenile, and adult. And the two regions that we chose was hippocampus and cortex to see if there was a difference. What we found is that injecting the cortex in pups has exactly what we have seen in literature in different studies, and so it's consistent. Where the injection, the cells will migrate so far up to 3 millimeters, as we can see here, across the entire cortex, and also and it will stand from the medial lateral position. And we can see when we plot it from the anteroposterior, we can actually see our injection site. We have, of course, more cells, but how they actually migrate so far. And important as a survival rate, we have 15%, which is in line with the literature. Many other papers have shown the same survival rate in pups. And the maturation profile in this case was also, as previously shown, most of them were PV positive interneurons and SST, somatostatin interneurons. However, when we injected in the cortex in juvenile and adult, and this is an example of the injection in the cortex, what we found is that the cells, they leave the injection site, but the distribution of the cells is mostly around the injection site. And we can see here in juvenile cortex that some cells, they migrate up to 600 micrometers from the injection site, but most of them, they will be around the injection site. And the same thing for the hippocampus. And this is an example of the hippocampus. This was the injection site. The cells will migrate and we will have some cells in the dentine gyrus, but the distribution now far and the migration, therefore, it will be mostly located in the around the injection area. But more important is that not only the migration is reduced, but also the survival rate is low. Maturation profile instead is always preserved. So how can we interpret those data and why do we have such different wide range? So if we had our results in the literature findings, we can see that there are two main groups of findings. There are groups in which we have a high survival rate and migration, and a group in which we have a decreased migration and survival rate. And I think that the difference is in the underlying circuit that we are going to treat. In particular, where we have an increased survival rate and increased migration, are all underlying circuit and recipient that they have an increased hyperexcitable circuit. We are talking about pilocarpine animal model, a TBI, a CCI, or a PAPS, and the P2, P4. To conclude this part, I will say that what we think is that when we use the MG cell transplantation, how the cells and the features of the cells will depend upon also the recipient animals that we are going to treat. And in particular, age and brain region and the disease state may play a role. So as an interpretation, we need to use the MG cell transplantation in a wider range of conditions, and to precisely modify the circuit, we need to think that if the circuit is less hyperexcitable, is the balance between excitation and inhibition is not perfect, but is more preserved than other condition, then we need to modify the protocol in order that we do need to have a distribution of the cell injected into the target circuit. So if the cells are going to be just located in one area, the protocols need to take in account, number one, how many cells I'm going to transplant, number two, how many injections I need to cover the area that I am going to study. And a single injection site will not work in that condition. And more important, because the different circuit that I am going to study and we are going to inject will have a different balance and a different parvabamine and somatostatin interneurons, is important to see in relation to that circuit how many more cells I am going to add, and that will make in literature easier to follow what really we need to change to have a translational purpose. With that, I would like to thank all the people in my lab, in particular Josie, who was a graduate student from Brazil, who helped me in the in vitro work that I present today. Tina and Harrison, they were a technician and helped me with all the background work to do the experiment, and Caroline, who helped me in the maintaining colonies and the MG transplantation. And of course, Scott, who was super supportive on these projects. Thank you, and I take questions.
Thank you so much, Rosie. That's really wonderful talk. I really enjoyed. So if anybody has questions, you're free to unmute yourself and ask questions, or you can type into the chat box and I can present it to the speaker. Oh, okay. So please.
I have a question. Hi, Rosie. Great job.
Thank you.
I'm wondering about the first part of your talk. So I think to support the idea that the hippocampus plays an important role in ASD, I'm just wondering, is it conceivable to also follow up the work with a hippocampus-specific knockout of CNTNAP2? And then, so if you see that the behavioral defects still exist, and in turn, if the input changes still exist, I'm just sort of wondering what your thoughts are on the cell autonomous versus the non-cell autonomous contributions.
That is a really good point, and I think there is not available yet the possibility to precisely knock out a specific region, because we do not have the flux on this animal model. Many other labs are, I think, looking for that because that will give the answer how alteration in a specific cell type will change that, and what contribution during development. It will be much more, it will add so many more information in how the circuit is changing.
Okay,
yeah.
So probably you haven't done a lot more detailed cellular level, like rabies virus tracing, just to see how these changes in the inputs, do you know what they're targeting, like which cell types?
No, I have not specifically done that type of work. I am working on trying to understand how parvabamine interneurons are playing the role specifically, just focusing on the parvabamine interneurons while they're performing the task, using different technique in single units and in 2-photon imaging. But those are as a future project so I don't have the result yet. But it will be really nice to see how that play a role while the animal is performing the task, and if that will change during the encoding or the novelty when the novelty signal is higher. So as a future, yes.
Thank you.
Mm-hmm.
Okay, probably Bobby.
I have a question, Rosie. So this is really, really good and a lot of work. I can tell how much work this is. So I had a question about the in vivo recording. So you're recording from CA1, and you're saying EC for enteral cortex input, and you have the slow gamma and the high gamma. But EC, as you know, I'm sure it gets separated into MEC and LEC.
Mm-hmm.
And how are you thinking about that in terms of CatNap2 and some of the changes that you're seeing, and where your recording electrodes actually are, right? So proximal CA1 will get a lot of MEC input, but distal won't, and vice versa, right? Distal CA1 gets a lot of LEC input. And so what do you think about that?
I think that, first of all, the recording is from the dorsal hippocampus. And so what we see here is one part of the entorhinal cortex input. The entorhinal cortex input is decreased because we have a reduced mid gamma, and this is across all animals, is very stable signal. But what we record in CA1 is a combination between the input coming from entorhinal cortex, but also the local circuit, how the local circuit is receiving the input coming from entorhinal cortex. What I know from this work is the local circuit is impaired. And I think the fact that the local circuit is impaired can be seen through different electrophysiological signal. For example, at the in vitro level, we have a reduced inhibition, but also the fact that we have a specific reduction in shortwave repulse power, it indicates that the pyramidal cells and the inhibition in pyramidal cells is impaired. So from this work, I know that there is an impaired inhibition locally. For the input coming from the entorhinal cortex, that plays a role, but also because it's a neurodevelopmental disease, I don't know yet if the input coming from the entorhinal cortex by itself is impaired. That's why, as in my future direction, multi-regional recording will help me to understand that.
Can I ask another question about the MGE transplants? So I'm really fascinated that depending on the age, and you said the brain region and also certain disease models, that these MGE cells do different things. And I'm wondering, like, how do you get at thinking about the mechanism? So is it feasible to at one day put them in, let them do their migration thing, maybe integrate or not integrate, and then maybe sort them out again and just see maybe by single cell transcriptomics or something to see if there's some influence of the environment on these cells to be able to work backwards towards a mechanism?
I think that what I see now is that it seems that excitation level in the circuit is playing a role. Those cells are moving more in a hyperexcitable circuit. And also, if in a normal condition, when there is no hyperexcitation, but in normal condition, when I transplant in the CA3, those cells move more to the dentate gyrus than to the CA1 area. So the dentate gyrus seems more receptive on taking the MG cells that are transplanting more than CA1. Yes, there are possibility in how to modify that and what actually we can target to have our goal that they need to migrate and how many of those cells will survive. We were thinking different ways to do that. I hope that there I can do some additional work to improve and to translate that in circuit where they are not migrating as we want.
Jenny, can I jump in also? There's a couple other factors the way we think about it. I mean, one, the first part of your question, obviously because the cells disperse widely, it's hard to get them back. So, I mean, from a technical point of view, you have a good question of studying them, but in single cell RNA-seq. But you know, you're talking about finding, you know, a bunch of randomly scattered into neurons, if you think about it. The other kind of way I look at it, at least, is two kind of key things from Rosie's work. One, obviously, and this goes to a lot of the interneuron development field, you know, the activity in the host brain plays a big role. You know, so integration and functional integration of these cells, you know, is influenced by the excitability level. And these are wild type animals, both young and old, cortex, hippocampus. So they're very different kind of parameters. And I think that's something that we'll be able to tease out. But the other thing I want to point out, and I think Rosie's, you know, polite enough not to do this, but I guess I'm not. The papers that have less than 5% integration, none of those are from my lab or group. All of the ones with 15% and better MG transplants are from our lab or Bobby's lab. So labs that were initially trained to do the MG transplants. And as you can imagine, it's not a trivial thing to just take out embryonic tissue from a, you know, day 35 mouse and transplant it. And so I think unless there's some standards of what actually is an MG transplant, it's hard to make these direct comparisons between labs.
Yeah,
And that's what we're hoping
right.
to do with Rosie's comparisons of historically our work, which replicates, you know, different people in the lab over different generations, as well as other labs that claim in their publications to have transplanted MG cells.
Okay, thank you. Anybody else has question? Ethan?
Yeah, as a follow-up to that, I'm curious if you see any differences, or if you've done it in like the CXCR4, CXC, what is it, 12? I think 11 or 12. In terms of those in the different disease brains, because I know that can control interneuron migration.
You refer to the transplantation?
Yeah, on the transplantation.
No, I have not. We have not done that work. Yeah.
Okay, anybody else has a question? So I have one question. So I'm very curious. So the PV interneurons, or you found less PV interneurons, or the PV interneuron density in the hippocampus in the mutant mice. And did you check on the SST? And do SST has any role in this local circuits neural activity? And the reason I'm asking is that during the development, I think interneuron progenitors migrate towards the cortex, and then at the cortex, BMP signaling plays a role in differentiating them into either PV or the SST interneurons at the expense of each other. So I wonder if it's coming from that developmental specification issue or the migration issue. And if you look at the SST, there might be some sort of answer to it.
So to respond to the first question about if we looked at SST, there is a work in literature looking at SST as a density, and they did not find any difference. But in this case, for this project, actually, it will be really nice to see how, even though there is not a difference in the density of the SST, it will be nice at how functional they play in correlation with PV during what we have seen. So we are actually growing the line to do that, to precisely target SST. To answer the other question, there has been done some work trying to understand if there is a migration problem. In this animal model, there are some migration problem, but that was not correlated with the interneurons. What they think is that the problem is on the maturation process of the interneurons. They seem to reach properly, but there are not many papers, there are just one or two focus on that. They reach the final target, and then it seems that there is a disproportional maturation of the interneurons.
Okay, thank you.
Mm-hmm.
Any other questions? Okay, thank you so much, Rosie, for a wonderful talk.